to do a superiority trial to demonstrate, okay, their efficacy is way beyond the things, and probably they never needed a trial. Then came the era of the superiority trial where we demonstrated efficacy over placebo. And now we are in an era where we have, barring some of the diseases, okay, where we don't have effective treatment, for many of the disease conditions, we now have effective treatments. And whenever a new drug or a device or a biologic that comes, it is very difficult to demonstrate efficacy over an already existing treatment which is efficacious. So here comes the, I mean that brings into this design, and now we are almost in an era of non-inferiority trial designs. Having said that, the regulatories have also increasingly understood this, and they also approve this as a part of, I mean, this is one of the pivotal clinical trials, you can say, to approve a new therapy or a new diagnosis. So therefore, this is one topic we thought that is going to be very interesting, and I now invite Dr. Anand. So Dr. Anand is an associate professor of clinical pharmacology. He is working in AIIMS Bhubaneswar, and his areas of interest are pharmacometrics and advanced clinical trial design, artificial intelligence, use of artificial intelligence in clinical trials. He also does a lot of clinical trial simulations and also precision medicine. So having introduced him, may I invite Anand, you to please share the screen and start the lecture.
Before starting the lecture, thanks, madam, for introducing me. And like Smita ma'am is and was and is my faculty in PGIM at Chandigarh. When I was a resident there, I was learning a lot under her. So it's definitely an honor that
we are learners.
If a teacher introduces a student in a forum like this. Thanks, madam. And I also thank Dr. Melvin for inviting me to deliver a talk on this. Thank you. And thanks, Geetanjali ma'am, because being a director of the institute, she is really busy in her schedule, and still she took out time to come here. And I'm really thankful for that. And I'm also thankful to all the participants who have taken their time to listen to this talk. Anyways, I will start sharing my screen and will proceed to my talk. Yeah. So, like, you can see from the topic itself, like it's called understanding the nuances of non-inferiority clinical trials. So as already Smita ma'am told about the non-inferiority clinical trials, like why it's important these days and the number of clinical trials have come up. If you observe actually the various publications on PubMed, where the clinical trials report a non-inferiority design, we can observe that the number of non-inferiority trials done are more in the pharmaceutical industry rather than academics. Okay. So it's not that non-inferiority trials cannot be done in academics, but I would say that it is the paucity of the information available which prevents the academicians to conduct non-inferiority clinical trials. So I am here just to demystify that. I have put the talk in a simple manner, okay, so that at the end of the session, I hope the students are encouraged to take up this non-inferiority design in their projects. Okay. So if we analyze the flow of our topic, first we will look into what is hypothesis testing. Then what is non-inferiority? How is it different from superiority? How is the sample size calculation done for non-inferiority studies, clinical trials, and how to analyze and interpret the non-inferiority clinical trials. Okay. Now, this is the forum where the audience do not need a definition on a clinical trial. Still, I will go ahead. Okay. So this is the definition provided by National Institutes of Health. Okay. To be simply put, what happens in a clinical trial? We, recruit the eligible participants and then assign them to various treatment arms and then analyze the effect of the treatment. So that's what the National Institute of Health have said. That is, a clinical trial is any research study that prospectively assigns human participants or groups of humans to one or more health-related interventions to evaluate the effect on health outcomes. Okay. Now, what is a goal for a clinical trial? Goal of a clinical trial? Well, there are many goals. So one of the important goal is hypothesis testing. See, other goal, you can tell that it's about the estimation of various parameters, but one of the major goal is the hypothesis testing. Okay. So what's a hypothesis testing? Let us look at hypothesis testing from a statistical point of view. Okay, so hypothesis testing is a form of statistical inference. Usually, when we design a clinical trial, okay, there will be this two terms, that is null hypothesis and alternate hypothesis. Okay, and usually our alternate hypothesis will be the desired objective of the study. Okay, like we want to see whether a drug works or not against a disease. The alternate hypothesis will be yes, the drug works in a disease, to put simply. Okay. Now, when we design a clinical study, the study always starts assuming the null hypothesis. Okay. And at the end of the study, we get the results and we analyze. So to agree with the alternate hypothesis, we will calculate the probability of that null hypothesis being true. Okay, so whatever the probability which we obtain after the data analysis in a clinical trial is nothing but the probability of the null hypothesis being true. And if that probability is very low, that is usually it is taken as less than 0.05. I mean, there is a reason why the 0.05 is chosen, but I'm not going to discuss about it. So if the probability of the hypothesis testing of the null hypothesis is less than 0.05, then we agree with the alternate hypothesis. Okay, right. Now, when we look into the various designs of hypothesis testing, there are three major types: superiority testing, superiority hypothesis testing, non-inferiority hypothesis testing, and the equivalence hypothesis testing. Okay, so in this seminar, I'm not going to talk about the equivalence hypothesis testing. Okay, I will majorly focus on non-inferiority, but I will also touch upon the concept of superiority, because to understand the non-inferiority, we need to understand the superiority also. Right. I'll start with superiority and then go to non-inferiority. Okay. So what are the scenarios in which we usually design a superiority trial? Okay. In the introduction part only, Smita ma'am told that, like insulin for diabetes compared with placebo. Okay. So usually such type of trials are superiority trials. Okay. Any placebo controlled trial will be superiority trial. And sometimes the superiority trials are designed against an active comparator also. Okay. The classical example being sofosbuvir in hepatitis C. Okay. So it's much more, what to say, it was much more effective than the drugs like interferon and ribavirin. Okay. So that is one situation where superiority design will be suitable for an active comparison, but usually it is always placebo comparison studies. Okay. Now, if we talk about the null and alternate hypothesis for superiority trials, they take the classical definition of the null and alternate hypothesis. That is, null hypothesis for a superiority trial states that both the clinical trial arms are similar, whereas the alternate hypothesis states that both the study arms are similar. Okay, right. So whenever we design a clinical trial, we have a primary objective. Okay, and let's say that a primary objective being the decrease in systolic blood pressure. Okay, right. So we have two treatment or two arms, I would say. One is the standard treatment plus placebo, and the other is standard treatment plus the new drug. Okay, so we get at the end of the study, we measure the fall in blood pressure in both the arms, and so calculate the difference between them. Okay, so we get something called mean difference, which measures the efficacy of the treatment arm. So apart from the mean difference, we will be also calculating a confidence interval. Okay, in a superiority trial, we will see what is the probability of the confidence interval crossing the midpoint. Okay, right. Let's say that this small line indicates the midpoint. Okay, here 0 for the parameters where the difference is measured, or 1 for the parameters where ratio is measured. Okay, so that is 0 for mean difference and 1 for odds ratio, something like that. Okay, now let's say the plus indicates the experimental arm, and the minus indicates the the control arm. Okay, so these are the various scenarios which can get at the end of. Okay, so yeah, let's say this white dot being the mean difference, and this arrow represents the confidence interval. Okay, right. This first scenario clearly indicates that the experimental arm is superior. Say, for example, the mean difference was 5 and the confidence interval was 3 to 7. Then it clearly indicates that the experimental arm is superior. Okay. So this scenario indicates that the experimental arm is inferior, or the control arm is superior. Let's say that the mean difference here will be -5, and the confidence interval will be from -7 to -3. Okay, but when it comes to the middle scenario, we cannot conclude that both the treatment arms are similar. Okay, we say that the treatment arms are inconclusive because the study is not powered sufficiently enough to detect the difference. Okay, so this is the issue with the superiority studies. Okay, there is a lot of if the study is not powered properly, the results will be inconclusive, or if the treatment is not working, then also we will get inconclusive results. It's difficult to differentiate between these two. Okay, so that is one of the reason why the non-inferiority is better than superiority studies. Okay, right. Now this is an example from for a superiority design. Okay, so here this is the PM idea of an article where the that is the pharmacotherapy, that is niraparib plus bevacizumab was compared against niraparib alone for platinum sensitive recurrent ovarian carcinoma. The title itself clearly says that it's a superiority trial, because if you observe the design here, it's a add-on placebo control design. Okay, so here we have niraparib in both the groups, whereas in the experimental arm we have bevacizumab, and in the control arm we have niraparib plus placebo. Okay, right. Now, if you observe the results, okay, here you can see that the result is an hazard ratio. That means if the confidence interval crosses 1, that means what to say, the superiority is not demonstrated. If you observe the results, the confidence interval for the adjusted hazard ratio for this niraparib plus bevacizumab arm is 0.35. That is the adjusted hazard ratio is 0.35, and the 95% confidence interval is 0.21 to 0.57. Okay, so this is how we interpret a superiority study. Okay, now this is how we calculate the sample size for a superiority study. I'm not going to explain much about it. You can observe that we have this Z value, which is the standard normal distribution value for alpha error, for beta error, and we have variance in the numerator, and the denominator we have the effect size. Okay, just remember this, because we will be comparing it against how to calculate the sample size for a non-inferiority trial. Okay, now we will come into the actual topic, which is nothing but the non-inferiority trial. Okay, now put simply, okay, what's a non-inferiority trial? Okay, here that is a trial where the experimental arm is literally non-inferior to the control arm. Okay, that is a clinical trial comparing an experimental arm with the control arm to determine whether the experimental arm is non-inferior to the control arm. Okay, so that will be the objective of a non-inferiority study. Okay, so what does the term non-inferior mean? Okay, so this textbook of design and analysis of non-inferiority trials by Rothman et al. States that if the effect of an experimental therapy on an endpoint is either better than or not too much worse than the effect of a control therapy on the same endpoint, the experimental therapy's effect is said to be non-inferior to the effect of the control therapy. Okay, again, let's take the hypertension example here. Okay, right? So we have the what to say, an experimental therapy. Okay, and we have a control therapy. Okay, let's say that the the control therapy, that is the control arm, reduces the blood pressure by say 8 millimeter mercury. Okay, whereas the experimental arm reduces the blood pressure by say 12 millimeter mercury. Okay, here I'm just discarding the confidence interval principles. Here, just by observing it, you can tell that okay, this experimental arm is working better, it's fine. But how about instead of 12 millimeter mercury, if the experimental arm reduces the systolic blood pressure by 7 millimeter mercury? Of course, it is lesser than 8 millimeter mercury from the control arm. Okay, but clinically speaking, there is no much difference between these two arms. So in this case, we can say that the experimental arm is non-inferior to the control arm. I'm just putting it generically. We cannot just conclude like this without the confidence intervals. I'm just giving an example here. Okay, so So we will see why these non-inferiority trials are essential in the forthcoming slides. Okay. Now, just like the scenario which we saw with the superiority studies, if we analyze the scenario for non-inferiority studies, almost always the non-inferiority designs are all what to say, active comparison designs. Okay, it's always the experimental arm versus active control. You will hardly find a study where a placebo control is used unless the placebo itself is found to be effective in treating a condition, usually in psychiatric condition, but still it will be very rare to observe clinical trial where a non-inferiority has been designed against a placebo. So if we take the scenario for a non-inferiority study, it's always experimental against the active control. Okay, it's like two active therapies are compared against each other. Okay, why non-inferiority trial? Yeah. So in the introduction parts, Tamam has already explained. I'll just repeat them again. Okay, like if there exists a standard treatment, usage of placebo is definitely unethical. Okay, right? It makes sense. You cannot deny a treatment to the patient just because a clinical trial is done. Okay. Now, here, as I said previously, when we talk about an active control study, unless the experimental arm is experimental arm that is experimental therapy works very well, it is very difficult to prove the superiority. Okay, so that is why you will find the non-inferiority designs more in the industry sponsored studies. If the industry sponsored studies or if the regulatory studies are designed to be superiority studies, then they can never get an approval for their treatment. Okay, they'll be spending, the companies will be spending crores of rupees or billions of dollars, and end up with study where they were not able to prove that their drug is working compared to the other. So that is why if we find even in the PubMed, most or almost all of the non-inferiority studies are the regulatory studies, industry sponsored studies. But this is one of the objective here, where I just want to show how the non-inferiority trials can be done in a academic setting also. Okay, I'll also share my experience in designing the non-inferiority studies. Okay, so that it's not a I would say that it's not a big thing in conducting a non-inferiority study in the academic setting also. Okay? And I might be wrong also. Nowadays many of you might be doing. Okay, I'll be happy if I'm wrong. Yeah, so this is, I'm just saying that doing a non-inferiority trial is pretty easy. Okay, now why non-inferiority trial, and so why do we require a new treatment when there is an already existing treatment? Okay, why? There are various reasons, and as a pharmacologist, you must be knowing all these reasons. I'll just list out the reasons. Like better safety. Might be the new treatment is having specific target. Okay, just targeting one specific receptor compared to the the existing therapies, so that it has a better safety profile. Okay, or let's say the newer therapy can be administered orally. Okay, whereas the existing therapy is administered parenterally, or newer therapy has a better dosage regimen. That is, the newer therapy can be administered once weekly, whereas the existing therapy has to be administered daily, something like that. Okay, so let's say that the newer therapy, let's take this second example. The newer therapy where a oral formulation is developed. Okay, so again, if we take the hypertension, of course, none of the antihypertensive drugs these days are, uh, administered parenterally unless emergency use is required. But just for the sake of example, I am telling. Okay, say that the existing therapy is administered parenterally and reduces the blood pressure by 10 millimeter mercury, whereas the newer therapy, which is administered orally, reduces the blood pressure by say 8 millimeter mercury. It's fine. We can go for the orally administered therapy. Okay, so that's why the newer treatments are developed, and that's why non-inferiority designs are required to evaluate the newer treatments against the existing standard treatments. Okay, right. So before going into the concept of the null and alternate hypothesis, we need to understand the concept of non-inferiority margin while designing a non-inferiority clinical trial. Okay, right? So the non-inferiority margin, again, this is a textbook definition from the same design and analysis of non-inferiority trials by Rothman et al. Okay, so that is the acceptable amount for which the experimental therapy may be worse than the control therapy and still be non-inferior to the control therapy is called a non-inferiority margin. Okay. Right? So this concept I have explained it previously also, but I'll explain it again. Okay. Let's take this. Let's take our antihypertensive example again. Okay. Let's say that this white band is a uh, effect of the treatment arm. Okay, let's say that we have an antihypertensive drug A, okay, which was evaluated against placebo in a previous study. Okay, and let's say that there was a reduction of 12 millimeter mercury. That the mean reduction of blood pressure was 12 millimeter mercury in the treatment arm of the previous study, and let's say that the mean reduction of blood pressure was 2 millimeter mercury in the placebo arm. Okay, see, right? Now, if we actually see the effect size, it is the difference between the fall of blood pressure in the treatment arm and the placebo arm. Basically, here it is 12 minus 2. So here the effect size for the treatment arm is 10. This is for drug A. Okay. Now, this drug A is an already established drug. Okay. Now we have another drug B, which is a newer therapy. Okay, and we want to prove the we want to prove that the drug B is non-inferior compared to drug A. Okay, right? So now we know that the effect size of drug A is 10. Okay, right? Let's say that this drug B has a better safety margin and has a better dosage regimen. So the drug B is non-inferior if it example 2 is the non-inferiority margin. Here, if we observe that non-inferiority margin of 2, it is 20% of the effect size of the control drug. Okay, right? There are no hard and fast rules for this, but the certain FDA documents recommend that 50% of the effect size of the control arm. Okay, right? I mean, there is another conservative approach also this delta which I have mentioned, that is the non-inferiority margin. So basically what we are saying that is, if the difference in the between the treatment arms is greater than the non-inferiority margin, then we can agree with the alternate hypothesis. And the opposite becomes the null hypothesis. That is, the difference between the treatment arms is less than the non-inferiority margin. Okay. So this one I will also reinforce this concept again in the forthcoming slides. So just remember that in a non-inferiority study, the null hypothesis will be the difference between the treatment arms will be less than or equal to non-inferiority margin, whereas the alternate hypothesis will be the difference between the treatment arms is greater than the non-inferiority margin. We'll come to that. Okay. So just like the various scenarios which we observed in a superiority study, we will also observe the various scenarios in a non-inferiority study. Okay. Now, let's say that let us assume that this midline is the 0 point for a mean difference parameter. Okay. Here, since it's a non-inferiority study, we will introduce a non-inferiority limit. Okay. Okay. Since then the limit favors actually the control arm, we are putting the non-inferiority limit on the control arm. That's how it is done. Okay. Now we will look into all the scenario. Okay. If we take the last scenario here, it's inconclusive because the confidence interval actually crosses both 0 and this non-inferiority margin. Basically, this is a clear example for an underpowered study, even for a non-inferiority trial. Okay. Then we have the scenario where you can observe that lower the lower confidence limit has crossed the non-inferiority margin, whereas the upper confidence limit has actually not crossed the 0 point. So basically, this scenario says that the clinical trial is inferior. So we can claim inferior because the non-inferiority trials are always sufficiently powered to detect the superiority or inferiority. Okay. And the first scenario, it says that it's clearly superior. Okay. Now, if we look into the second and third scenarios, you can clearly observe that the lower confidence limit has not crossed the non-inferiority margin. Okay. So these two studies are non-inferior. Okay. I'll come to an example. Okay, but just see that the second and third scenario are the perfect example for a non-inferiority study results where the alternate hypothesis is agreed. Okay, and interestingly, you can see that in the third scenario, if we take this 0 point, it is actually inferior, whereas if we take the non-inferiority limit, the experimental arm is actually non-inferior. Basically, since the study was designed for a non-inferiority study, we can always conclude that the treatment arm is non-inferior rather than the treatment arm is inferior. Okay. This is an example for a non-inferiority study. Okay. Here you can see that this newer drug, lenvatinib, which was compared against sorafenib, which was it is the standard therapy in first-line treatment of patients with unresectable hepatocellular carcinoma, non-inferiority study. Okay, so here, as we saw previously, for designing a non-inferiority study, we should always define a non-inferiority margin. Okay, here the non-inferiority margin defined was 1.08. Okay, right? Because you can here, if you observe the result, you can see that the median survival time for lenvatinib is 13.6 months, whereas for sorafenib is 12.3 months, and you can clearly observe that confidence interval, that is this 1.06, has not, what to say, crossed the 1.08. Okay, so here in the class, in the previous slide, you can remember that actually it's the, what to say, lower limit should not cross the non-inferiority margin, but in this example, because we are measuring the hazard ratio, it turns the other way around. Okay, so basically here the upper limit should not cross the non-inferiority limit. Okay, so this clearly proves that the study, the drug is non-inferior. But you can also observe that what to say, the confidence interval is crossing the midpoint, which is 1 here. Okay, so we cannot claim here superiority, but it clearly proves that the new treatment is non-inferiority. So that's what, again, see, this is a regulatory study. This is a regulatory study. If the what to say, sponsors had designed for a superiority trial, then the result would have been inconclusive, and the regulators would not have granted approval for marketing the drug. Okay, whereas here you can clearly observe that even though the drug is not superior to the existing therapy, the drug has been proven to be non-inferior. Okay, so that is how the non-inferiority design or the non-inferiority trials are useful compared to the superiority design. Okay, right? Now, when we talk about, if we talk about the sample size calculation for non-inferiority studies, if you remember from the sample size calculation formula from the superiority studies, it looks very much similar, except that there are two differences. Okay, one is here, if you had observed, it was alpha by 2 for a superiority design, whereas here it is just alpha. Okay, because we are concerned about only the lower limit of the confidence interval not crossing the non-inferiority margin. Okay, so that's why we are performing a one-sided test here, and in the denominator we have the non-inferiority margin along with the effect size. Okay, but almost all the studies, this effect, the difference in the what to say, the endpoints between the two arms are assumed to be 0. Okay, the difference is assumed to be 0. Okay, here even in the previous example where calculating sample size, definitely the what to say, they would have assumed that while calculating the sample size, it was assumed that there is no difference between the two, only the non-inferiority margin was considered for calculating the sample size. Okay, so this is just to complete the mathematical formula. They have introduced this mu, what to say, the mu a minus mu b. Otherwise, in the denominator, usually there will be only non-inferiority margin present for calculating the sample size. So you can observe the same thing here also, right? You will be having the that is, this is for sample size calculation where the endpoint is categorical. You can observe that in the denominator we have non-inferiority margin, and in the numerator, instead of alpha by 2, we have just the alpha. Okay. Now, right. Okay. So I have troubled you a lot with the theoretical part. Now I'll just what to say, show some examples and share our experience in designing a non-inferiority study. I mean, I'm not going to discuss the entire design, just did bits of it, just to explain that doing non-inferiority study is actually not difficult at all. Okay, so I'm just exiting and I will go into this window. Right. Okay. So here, I'll be just demonstrating the calculation of the sample size calculation of sample size for a non-inferiority study. So this is many of you might be aware this interface. Otherwise, this is the R Studio interface which runs the R programming language. Okay, and the again my hidden message here is, yes, the pharmacologists actually should learn any of the programming language concerned with the data analysis. Okay, I would say that for the budding pharmacologist, this is not a luxury. This is an essential talent now, or essential skill now. Not it's an essential skill now. Okay, so that it will be definitely helpful in the future endeavors. Okay, so nowadays the programming languages which are majorly used are either R or Python, and definitely the importance of Python is growing more than R, but both has its own advantages and disadvantages, and it will be better if you learn at least these two programming languages, that is R and Python, and perform data analysis with that. Okay, right. So here I'll be not explaining the syntax in R, but you can understand what is being done here. I'll just clear this stuff so that it will be easy to understand. Okay. Now I'll explain the sample size calculation for a superiority study so that we can understand this calculation of sample size for an inferiority, a non-inferiority study. Okay. Here, no need to explain all these parameters, because you already you know that this first parameter is the alpha error, the second parameter is a beta error, and this is the pooled standard deviation, and this is the allocation ratio, and this is the the effect size which is present in the denominator. Okay, so basically these are the various parameters to calculate the sample size for a superiority study. I'll just run this code. You can observe that to measure a difference of 5 between the two groups with a pooled standard deviation of 10, we need a sample size of 62 in each group. Okay, here the alpha and beta errors assumed are 5% and 20%. Okay, now you can see that the same thing. I mean, we will be using a similar strategy to calculate the sample size for a non-inferiority study. Here, this here equality means here it's a superiority study. That's what the code, that's what the code means in this command. Here, NIS is either non-inferiority or superiority margin. Okay. Here also we punch in these same values for alpha and beta errors, which is 0.05, 0.2. Okay, the pooled standard deviation to be 10, the allocation ratio to be 1. Here, as I told previously, the difference in the effect size is always assumed to be 0, whereas I have defined a non-inferiority margin of -5. Okay, right? You can observe that the sample size here is 49 per group. Okay, right? But theoretically, when you all read an article or get into a discussion, there will be always a discussion that for a non-inferiority study, the sample size is always more compared to a superiority study. But here you can observe that actually, if we punch in the same parameters, okay, I didn't alter any parameter, okay, right? But still we have got the the sample size for a non-inferiority study to be lesser than the superiority study. Okay, right? So it's actually not correct to compare the sample size calculation between the superiority and non-inferiority study design. Actually, it's comparing like, classically everyone will say that it's like comparison between apples and oranges, that you cannot compare, okay? Because the objective of a superiority and non-inferiority studies are entirely different, okay? You know why mathematically the sample size here is less? Because non-inferiority study, the test done is always one-sided, whereas here for a superiority study, the test of statistical inference done is two-sided. So that's why the sample size calculation for here is more for superiority study compared to this non-inferiority study when all the other parameters are same. Okay, right? Now here, this was the actually sample size calculation in one of our department's thesis, where there were two drugs comparing. The end of the study measured the reduction in VAS. Okay. So here for our study, we assumed again the same thing: alpha to be 5%, beta to be beta error to be 20%, the pooled variance to be 0.7, allocation ratio to be 1, the non-inferiority margin to be 0.35. Okay, here see, if you observe here, I have put minus in front of the non-inferiority margin because this code also allows the concept of superiority margin to be included for calculating sample size. To know about the use of superiority margin, there are many articles published on that. You can definitely refer to them, but usually still now superiority margins are not widely used. Okay, but if you are designing a study where superiority margin is used, then that should be also accounted for the sample size calculation. Okay, so I will not deviate much. So this was the sample size calculation for our study, and it came out to be around 50. Okay, so this is how we started with the calculation of sample size for one of our thesis. Okay, similarly, we have also helped the faculty in Department of Anesthesia to design their non-inferiority studies, starting from calculation of sample size to the analysis part also. Okay, right. I will come to the lecture part. Okay. So now that you would have observed that the calculation of sample size for non-inferiority study is not difficult at all. Okay. Now we will look into the the interpretation of the results from the non-inferiority studies. Okay, both analysis and interpretation, I would say. Okay, now let's say that there was a clinical trial design with two groups, that is A and B here. So we have two groups, A and B. Okay, at the end of the study, let's say that the primary endpoint was a continuous variable. Okay, for the group A, the primary endpoint mean was 14.14 and standard deviation of 6.03, whereas for group B, the standard deviation, the mean was 14.68 and the standard deviation of 5.59. Just by observing superficially only, we can conclude that there was no difference between the two groups. Okay, let's say now we will see if the results of these two groups, let's say that we will consider two scenarios. Okay, one is the superiority scenario and another is the non-inferiority scenario. Okay, let's consider first the superiority scenario. Let's say that these results were the product of a superiority study. Then if we analyze the results. That's if you apply the statistical test, simple t-test here, you can see that the confidence interval, the mean difference is minus 0.54 and the confidence interval is minus 2.85 to 1.77 and the p-value is 0.64. Basically here the null hypothesis is accepted. Okay, right? Basically the results are telling that there is no difference between the two groups because the probability of the null hypothesis being true here is 0.64. It is not low, it is not less than 0.05. So there is no difference between the two groups. Basically the study is inconclusive here. Okay, now let's say that the same results are being obtained from a non-inferiority study. Okay, so if for a non-inferiority study, we should always have a non-inferiority margin. Okay, let's say that the non-inferiority margin was -3. Okay, so again, now you will be all familiar with this diagram. Okay, this is the midpoint, the 0 point, and this -3 being the minus delta, which is nothing but the -3, which is the non-inferiority margin. And if we put the confidence interval here, okay, you can see that the minus 2.85 to 1.77 is not crossing the -3. That is minus 2.85 is greater than -3. So basically here we are telling that the newer treatment arm B is non-inferior to what to say, the treatment arm A. Okay, basically here we are accepting the alternate hypothesis here. Okay, so see, same result, but for a superiority design we accepted null hypothesis, whereas for non-inferiority design, same study, same results, here we are accepting the alternate hypothesis. For superiority you accept a null hypothesis, for non-inferiority you are accepting alternate hypothesis. So now you can understand how useful the non-inferiority trials are. Okay, right? But one of the during one of the discussions in the journal clubs, we came across this article. Okay, so you can see that it's a non-inferiority clinical trial. Okay, it's a non-inferiority clinical trial. You can clearly observe that the non-inferiority margin was -10%. Okay, right? You can see that the 95% confidence interval is from -10.5 to 5.3. Okay, right? You can see that the lower confidence limit is actually crossing this -10, but still the authors had claimed that the study was non-inferior. Okay, so like whenever we find the so-called mistake, I'll put the so-called mistake in quotes. Okay, because actually it's not a mistake here. We are like the lower limit is crossing the non-inferiority margin. How come the investigators are claiming non-inferiority, and it has been published in a journal of good repute, that is clinical infectious diseases. Okay, then when we started analyzing like what would have been, what we missed here, or whether they are wrong or whether our understanding was wrong, okay, then we came to a conclusion that actually it was our understanding which was wrong and the investigators were correct here. Okay, right? For that we need to, only if we know how the data is analyzed properly in a non-inferiority study, then we can understand what went wrong in our understanding. Okay, so for that, so this is a standard normal curve which everyone will be familiar here with. Okay, right? So on the x-axis we have the probability, it's a mean difference, and on the y-axis is the probability. It's not what to say, x-axis it's wrongly marked, it's the mean difference. Okay, right? if you remember from the previous slides, I told that in a superiority design, that is what to say, it's the probability of the results crossing the zero mark. That's the probability which will be calculating for a superiority design. Basically, this will be the probability for a superiority study. Okay. Whereas for a non-inferiority study, it is the probability of the results crossing the non-inferiority margin here. Okay. Right? Okay. So basically, it's the probability of the result crossing -3 and not 0. Okay. Right. Now I'll tell you how to analyze the result and how to calculate the probability for a non-inferiority study. Okay, so basically these are the two tools which I found that which it can be useful for calculating or analyzing the non-inferiority study. At least the graphical user interface of SPSS is not useful when it comes to analyzing the results of a non-inferiority study. Might be the coding part of SPSS might help. I have not explored that, but I might be wrong also. But as far as I know, even that good statistical software like SPSS, I didn't find any option. And SPSS, I'm talking about the version 22 or 23. Okay, right now 26 is running. Okay, right. So now I'll tell you. I'll demonstrate the analysis part also. Okay, right. I'll not explain these codes. So we have two groups here, A and B. Okay, so the mean of A is again 14 point, the same results from the previous example. Okay, mean is for A is 14.14 and SD is 6.02, and mean of B is 14.68 and SD of B is 5.5. Okay, here you can see that for a superiority study we have used this two-sided analysis, and we have clearly mentioned that the mu parameter is 0. Okay, so you can observe that the confidence interval, of course, the p value is not significant, but if you can observe that the confidence interval is from -2.84 to 1.76. Okay, now if we analyze the results from the non-inferiority analysis, that is, you can see that instead of two-sided, I've used the greater command, and instead of mu equals to 0, I've used mu equals to -3. Okay, I'm not going to explain the other parts. Okay, it's not relevant to this discussion right now. Okay, now I'll run this. Okay, here you can see that p value is significant. That means for non-inferiority study, we are accepting the alternate hypothesis. But the interesting part here is, here the confidence interval starts at -2.84 for superiority study, whereas for a non-inferiority study, the lower limit of confidence interval is -2.46, which is greater than the lower limit for a superiority study. That is just because the test is one-sided. Okay, that's why whenever one-sided test is done, only one part of the confidence interval is always reported. The other part is always reported as it's usually not reported, or sometimes reported as infinity. Otherwise, usually it is not reported at all. And these days, I have observed that in many of the non-inferiority trials, while providing the results for if the primary objective was non-inferiority, they would never mention about the upper confidence limit at all. Okay, just because the test was one-sided, the lower limit was greater than the superiority study. So the same thing actually would have occurred in this study also. Okay, here this minus 10.5 to 5.3 was from the superiority analysis, whereas for a non-inferiority analysis, this would have been definitely greater than minus 10. Okay, lower limit. So that's why the authors claimed non-inferiority. Okay, so I mean, these are not the big things. I mean, I will not tell. I will not use the term big things. These are all not difficult concepts to understand. It's just that the ignorance of the way to analyze the what is the data prevents us from designing and analyzing the non-inferiority studies. Okay, this is actually very simple thing. Okay, especially if we know the statistical programming languages, it's not at all hard to crack these things. Okay, so that is one of the simple nuance of analyzing a non-inferiority. It's very simple, just that many of us are ignorant about it. Okay, right. So even like only this JC in our discussion prompted us to explore more, and then we found that how the non-inferiority studies are analyzed, and that's how we came to a conclusion that our conclusion was wrong while criticizing the study. Okay, right. So when we talk about the claiming superiority in a non-inferiority design, I will not talk much about it, but yeah, usually the non-inferiority study will be sufficiently powered for a superiority design also. So there is no harm in claiming superiority or non-inferiority, but the reverse is not true. We cannot do a post hoc non-inferiority analysis. Non-inferiority analysis is always pre-specified. Post hoc non-inferiority analysis is usually not encouraged. It's possible mathematically, but usually not encouraged. But still, there is conservative approach for Bonferroni corrections. Okay, fine. Now, just this slide I have put just to tell that our understanding, that whatever I have taught is just the basics. Okay, because the understanding of non-inferiority goes even beyond that. Okay, so this slide mentions about how the the random dropout will affect the non-inferiority analysis when the per protocol analysis is done, and when an intention to treat analysis is done, and if the intention to treat analysis is done by treating the missing values with the last value carried forward format, how the results will be. That's what I'm not going to explain much. Just you can observe that, for if you can see that in while analyzing a non-inferiority study, if the random dropout was 10% in this trajectory, okay, what to say, both the analysis were significant, whereas if the dropout was 30%, actually ITT was not significant, whereas per protocol was significant. Okay, so I have given the DUI. You can go and explore about these things when you have time. The conclusion about this slide is, whenever we do non-inferiority study or even a superiority study, if we are planning for both per protocol and intention to treat analysis, if you are planning for intention to treat analysis, it's always encouraged. It's always better to do both the per protocol analysis also, and show that both the analysis do not differ from each other, which will prove the robustness of our study. Okay, so I think that should be sufficient for now when it comes to the understanding the basics of non-inferiority study. Okay, so yeah, that's what we saw about the hypothesis testing, the scenarios for non-inferiority, how to calculate the sample size, examples of a non-inferiority study, and how to analyze the study results. Okay, so that's all about the non-inferiority studies. Thank you.
Thank you very much, Anand. Thank you for that excellent talk. And we'll take up the questions. Before that, Dr. Melvin, do you have anything to say or about the session? Or we actually had thought of a short sort of assessment how our participants did, and that's we have I mean it's not going to be one-sided, it has to be two-sided. So participants have to answer these questions, which is based on the non-inferiority trial concept. And may I request Dr. Melvin kindly start with the first question. And in the meantime, I will encourage participants to post questions for Dr. Anand, for me, or Dr. Melvin, anyone else. And basically, it could be subject-related question or comments, or you could also suggest, I mean, you can give a couple of suggestions to make this whole endeavor more successful of our academic and with the first question.
Are you able to see the poll questions?
Not really.
Yes, I can see. I can see
Okay.
the poll questions.
Okay, fine. So you can just scroll down after you start answering.
So, Dr. Melvin, I am unable to see the question somehow.
Okay.
Do I have to press something?
No, nothing. It should be seen, ma'am.
I'm able to see, but how are we supposed to answer?
Just click on the choice. There's an option, right?
Click on the round hollow rods. Yes.
Just
yes.
Okay.
click
So,
on choice.
should I read the question? So let me just read it out. What is the foremost reason for choosing a non-inferiority study design? The available drugs are not efficacious for this condition. The available drugs are unequivocal efficacy. The available drugs are marginally efficacious, and withholding them would lead to serious life-threatening conditions or disability. Or the available drugs are very efficacious, and it would be unethical to withhold it. So we have 10 seconds to answer this question. So, okay, everyone, can we move to the next question, Dr. Melvin?
Yes. Yes, you can see it.
Okay. Number 2. So typically the control in non-inferiority clinical trials include option A, placebo control, option B, active drug control, option C, historical control, option D, a double dummy control. Okay, so we will discuss the answers in the after we finish with the final question, or we will do it now, Dr. Melvin.
The option is like once you complete the whole thing, it will display. So we
Yeah, I think
all the 5.
We'll finish and we'll see the we will assess our participant, and then we will discuss.
Yes.
We'll quickly go through that. Okay. So number 3. So can you please project that?
Yeah.
Yes, are we there, Dr. Melvin? I am somehow unable to see the screen. I'm just reading it out from my laptop. So the aim of the non-inferiority trial is to prove that the efficacy of the new drug is at least better than the comparator by a specified margin, is exactly equivalent to the comparator, is not worse than the active drug by an acceptable margin, is not better than the active drug by an acceptable margin. Okay, participants?
Yeah, now the
Okay.
fourth can they can do.
Yeah. So yeah, next we are going to the question number 4. So you posted the question number 4,
Yeah,
Dr.
yeah,
Melvin?
yeah. It's there. All 5 questions.
Okay.
are there. They just have to scroll.
Okay, I see.
You
So
can
the
read it.
validity of the results of the I am somehow unable to see the screen
You
for
can
the question.
continue reading, ma'am. 4th one.
Okay. So the validity of the results of the non-inferiority trial is in question if the test drug group is found to be way superior to the control drug group. The test drug group is found to be way inferior to the control drug group. The control group demonstrates a magnitude of efficacy more than the previously reported trials. Or if the option D is the control group fails to demonstrate the magnitude of efficacy as compared to the previous trial in the same setting. So please answer this question, question number 4. Okay, so can we move to question number 5 now?
Yes.
So which of the following is not true about the choice of non-inferiority margin? So Dr. Anand very nicely explained this concept. So I hope our participants are going to give 100% right answer. So let me read out the options. It should be more than the effect of the standard drug over placebo. It is, make sure it is not true about the non-inferiority margin. So option A, it should be more than the effect of the standard drug over placebo. It should be less than the effect of standard drug over placebo. It should be a minimum of 50% of the effect of the standard drug over placebo. The smaller the margin, the larger is the sample size required to demonstrate the results.
Yeah, 29 have answered.
Okay, so there we go.
So yeah.
Okay, so can we have the results, please, Dr. Melvin?
Yeah.
So for question number 1, how many positive responses, I mean, correct responses we have? Percentage?
Can you all see this?
Okay, for I can now see the polls. Yes. Okay, so around 52% have answered the question correctly. No, sorry, the 32% people have answered the question correctly. D is the right answer is D. The available drugs are very efficacious. Therefore, it would be unethical to withhold the therapy. As Dr. Anand also said in the beginning, and I also explained, the premise of this non-inferiority trial is to explain or to demonstrate that they are as efficacious as the existing treatments are. So as I said that the like insulin and penicillin, we have possibly exhausted on the very, very efficacious ones. And now whatever we are getting, they are almost, or at least they should at least demonstrate that they are equi-efficacious. So most of the times the non-inferiority trials are designed because it would be unethical to withhold the effective treatment. So now let's go to question number 2. So question number 2, everybody, almost 90% of you have given a correct answer. So that's wonderful. So my job is done here. Now question number 3. Again, again, happy about it. Now question number 4, 55%. Now the right answer is D for this question. The validity of the results of the non-inferiority trial is called into question. So the answer is D. If we do not, actually this is again, Dr. Anand also mentioned this term, assay sensitivity. If this trial per se, your non-inferiority trial actually is not designed for demonstrating an assay sensitivity. So in 2016, actually FDA took out this document, how to choose this non-inferiority margin, and the concept of assay sensitivity, which was always questioned before this period, they incorporated this into it. So though our study does, I mean, this non-inferiority study, let's say, does not, I mean, it will not have a placebo group to demonstrate this assay sensitivity. Assay sensitivity essentially would mean that drug is better than no treatment. So that obviously you can't demonstrate in the non-inferiority trial. But if there have been previous study of standard drug or the comparator has demonstrated a certain quantity or the certain magnitude of effect, your study at least should demonstrate that much magnitude. So therefore, it is always considered that your study should be carefully planned so that you do not miss out on getting this magnitude of effect, or you should not get this, there should not be any omission in getting this magnitude of effect. So that's it. So that you can avoid this type 2 error. Okay, now let's move to the 5th question. Which of the following is not true about the choice of non-inferiority margin? So somehow possibly this was a confusing question. This is, I kind of mentioned not true, emphasized. Somehow this point was possibly, it did not percolate through very well. So the maximum people have answered C. It should be minimum of 50% of the effect of the standard drug. So I think people got it as true about the choice of non-inferiority margin. So somehow this is the standard, 50% of the effect of the standard drug over placebo. So the answer is actually A. It can never be more than the effect of the standard drug over placebo. So that's it. Thank you, everyone. And it was indeed satisfactory for me. I think last question, I made it a very confusing statement just to test whether you are awake or not. Okay, now over to Dr. Anand for the questions that the audience have asked. Okay, I'll now I'll see if there are more questions. I've noted the previous questions.
So there are not
Okay.
questions. You can start the questions.
Okay. So there are no new questions. So the first question that we got was from Dr. Noor. And he wants to know how to measure the confidence interval. So Dr. Anand, would you like to take it up?
How to measure the confidence interval? Yeah. So like
this is not.
It's a completely mathematical aspect, but there is no need to measure and all just by applying the statistical test. Any statistical tool will give the confidence interval, which I during the demonstration only I showed. Just run the test, it will definitely show the confidence interval. Of course, confidence interval mathematically depends on your standard deviation and the sample size, that is standard error, and tools just give like that. Otherwise, it's all about the hardcore mathematics, which, to be honest, I have to refer the books and then come again if you want.
Dr. Anand, when I was a student, MD student, we used to do all this by hand. I mean, we used to use our, in fact, I have done a meta-analysis with Excel. So at that time, we used to use that formula plus minus 1.96 into SE.
Yeah. Yes, madam.
So that's how we used to calculate. Now the softwares make our job really easy. So though that was not really related, but no question is a bad question. So I really appreciate your question, Dr. Noor. So it is mostly a mathematical calculation, and there's a formula to it. But almost all softwares actually take it out with the click of a button. Okay, let's go to the next question. So now the next question is from Samiksha. So Samiksha says, sir, for a sponsor, if the non-inferiority is the only target to launch a product in the market where already a standard of care exists, it might fail post-marketing also. Especially newer treatments are always costlier than the standard of care, and is not also the approach of the sponsor to the market to market the product. Okay, let us take it in fragments. So Samiksha essentially asks, it is very if a newer product, I mean a non-inferiority trial is conducted to launch a product for marketing, and there is always a standard of care which is existing, it might fail post-marketing because they are costlier also. So Dr. Anand, would you like to take this up?
Yeah, so like I'm not disagreeing to the fact, but it doesn't usually happen like that, because these drugs, as in the beginning only, as we told, it's the newer drugs are always developed for better improvement of quality of life, or quality of life as a whole, compared to individual efficacy endpoints. So if the people can afford the drugs, then it might not fail. Okay, like we cannot just come to a conclusion that it will fail if non-inferiority is used. Rather, it's the other way around. All the drugs nowadays are actually approved through non-inferiority design. Like, I hardly find a study where superiority design is used if an existing therapy is already available.
Right, I completely agree with you, Anand. I understand what I understand from Samiksha's question is, Samiksha, actually, I mean, it's a little complicated kind of equation. So when we talk of the science, non-inferiority is possibly essential a design to bring this drug into the market. And as Dr. Anand mentioned, that many a times all these are to bring in those small differences or small improvements in the current existing therapeutic armamentarium. So it could be better route of delivery, it could be better safety, etc. So now your question about the costly thing or the economic aspect of it, you're very right in pointing that, because the regulatory agency at that point, when it actually approves a drug, does not look at the cost. You're very right in saying that, because the regulatory is interested whether we are providing a little more than what is existing, or to say that okay, they are eco-efficacious to start with. So the complication actually starts when during this reimbursement process. So where there is economics involved. So the economics of it, if both the drugs are providing similar efficacy, one at a higher cost, then the insurance company or the reimbursement agency actually will go for the version or the option that we have for which is here. So it is complicated. Move on to the next question. Sir, can we have superiority? Can we claim superiority from a non-inferiority trial? This is by.
Yes, here actually I would like to combine the answers from 2 or 3 questions. Yes, we can claim. There is no harm in claiming superiority from a non-inferiority. As I already told, usually the non-inferiority trials are powered sufficiently enough to claim superiority. But there are some conservative investigators where they will be, you can say that deducting the alpha to claim superiority. Something like only the superiority will be claimed only when the p-value is less than 0.001, something like that, for the same endpoint. But in another question, someone has asked, can a study include both superiority and non-inferiority? Absolutely yes, but not on the same endpoint, in the sense that, again, there are studies where primary will be non-inferiority, whereas the secondary objectives will be superiority. But when we again talk about the superiority and non-inferiority on the same endpoint, yes, still we can definitely go for it, either directly or by deducting the values from the alpha errors. Yeah, it's possible, and I've seen many publications like that.
Yeah, these days now, I mean, they're kind of making it a seamless kind of a design in terms of statistics, actually.
Yes.
So you can infer based on the results that you got, whether your quantitative estimates, confidence intervals, as well as the p values that you get. And that, but then you actually take it into account before you start your plan your study, incorporate it in your sample size calculation, etc. Yeah, thank you, Dr. Anand. Now,
Yes,
can we move to the next question? How we can decide the non-inferiority margin prior to the study? Dr. Noor Hussain asks this.
Non-inferiority margin actually calculation, that is what, it's a collective effort, wherein usually we bring in the clinical specialists who are interested in that study, and we always discuss with them like how much can we sacrifice for this newer therapy, something like that. And also we can always look into the previous studies for the control group, and we will come to a decision. Okay, so I mean, from my own experience, if I want to tell, when we were designing a study for some anesthesiology studies, I mean, they were comparing 2 procedures of anesthesia. I mean, they told that, the investigator was that I cannot even allow a 5% for non-inferiority. The non-inferiority margin should be not more than 2%. And we went ahead with calculating the sample size for that 2% non-inferiority margin. But eventually, the newer treatment was not non-inferior actually, because the non-inferior margin was very less. But the investigator finally accepted, yeah, that is fine. I cannot sacrifice the safety just to prove that the newer treatment is non-inferior. The results are fine, and that result have been actually submitted for publication also. So that's how it's usually done.
Right. So there is one last question, Dr. Anand, and I think you have already answered this. Can a study design encompass both superiority and a non-inferiority design? What about the sample size calculation and principle?
A sample size calculation, of course, anything which is it's always calculated based on the primary endpoint. If the primary objective is non-inferiority, use the non-inferiority principle for sample size calculation. If the primary objective is superiority, then use the superiority concept for sample size calculation, as simple as that.
Right.
There's just one more question, ma'am. The last one. In case
Okay.
of psychiatric clinical trial, which trial design is better,
Uh-huh.
placebo-controlled trial or a non-inferiority trial design for psychiatric?
Correct. Yes, that's from our boy. Dr. Kumar Well is asking this question.
Which trial is best, like placebo-controlled trial or a non-inferiority trial for psychiatry? Again, see, there is no blanket answer for this. It depends on the case-to-case scenario. Of course, if we are comparing two, say, antipsychotic drugs in schizophrenia, it's better to go for a non-inferiority study. If we again go for placebo controlled, I mean, not placebo controlled per se, but add on placebo controlled studies. Okay, even though in psychiatric there is a proof that a placebo works, but practically, nobody gives placebo for the treatment, and the psychiatric co-investigator will not agree for a placebo controlled clinical trial for psychopharmacological drugs. So yeah, if it's an add-on design, go for a superiority design. If it's a active comparator design, go for a non-inferiority design.
We have one more question that I can see in the chat box. So this is from Dr. Arun. If we want to see absolute difference between treatment, can we go for placebo trials? If we want to find relative difference between treatments, non-inferiority trials, is it that way? He wants to know.
If we want to see the absolute difference between the treatment, if I understand the question properly, are you asking that should we have to design two separate placebo control superiority design and find the difference or something like that? Then if the study is ethical, we can conduct like that, but still we cannot just combine the results from those two study per se unless we perform a network meta-analysis. But if relative difference between the two, I mean, I don't here understand what the term relative difference means. But of course, if you want to compare the difference between the two treatments, definitely non-inferiority designs will be helpful, because anyways, at the end, we will be getting efficacy parameter along with its confidence intervals. Yeah.
Yeah, if I may add one more line to your answer, Dr. Anand. So even in the placebo control trial, I look at it that way, that even in the placebo control trials, you actually get the relative difference because placebo also has some effect. So that's what we want to kind of negate in this. I mean, we want to deduct that effect size. So almost everything is like a relative effect when you are doing a comparative thing. And about this relative difference, I'm little not sure. I mean, possibly he wants to know whether the non-inferiority trial effect size will tell you whether it's a relative difference between the two drugs. Yeah. So that's all. Thank you very much. And it was really a very interactive and engaging session. And I thank Dr. Melvin and most importantly to Dr. Anand for this excellent demonstration of R and also encouraging the participants to learn R and also making us believe that these are not trials. These are not trials that can only be conducted in the industry settings. In staying in academics also, we can conduct this kind of studies. And that's all from our side.
Thanks,
Hope
madam.
to have more sessions like this in future. And also Dr. Anand will. Thank you very much, Anand. Thank you so much.
Thanks, ma'am, and thanks, Dr. Melvin. Thanks, Dr. Bhaskar, and thanks everyone for attending the session. Have a good night.
Yeah, thank you, Dr. Anand, for
Thank you. Thank you.
Thank you, Dr. Smita, for moderating the session. And a big thanks to each and every participant. You stayed, it was 1 hour 40 minutes, but all of you stayed patiently till the end. Thank you so much for that. And there are some question about whether we are going to have some journal clubs. So we will be having journal clubs in the near future. If there's any resident who is interested, you can come forward and just share your details so that we will get in touch with you also. And looking forward to see you in the next clinical pharmacology seminar. Bye-bye. See you all. Good night. Thank you, Dr. Bhaskar, also for helping in moderating this evening session. Thank you. Thank you.
Hello, Dr. Melvin.
Yes, Bhaskar.
Yeah, just before we wind up, I just wanted to announce that tentatively the next session will be on 17th, that
Okay.
is Sunday. The speaker who is taking is going to talk on immunotherapy in cancer.
Okay.
So he has asked if any of us want any specific point to be discussed or any specific question to be addressed, so that he will incorporate that in the presentation.
Sure.
So I think all of us are in the WhatsApp group. So if we want any specific thing to be discussed, then we can put that in the WhatsApp group. I'll communicate it to the speaker.
Sure.
Yeah.
Sure, Dr. Bhaskar.
Yeah. Okay. Yeah, that's all. Thank you.
Thank you, Dr. Bhaskar. Thank you, everyone.
Thank you, everyone. Thank you.
Thank you, everyone.